Hi, welcome to Fun with the Chemistry Club, where we highlight local and regional scientists and their achievement towards the betterment of society. Today we have Dr. Thea Scantlebury-Manning, a biochemistry lecturer at the University of West Indies Cave Hill campus. Hi, and good evening, Dr. Manning.
Thank you for inviting me.
Yeah, the pleasure is mine. Thank you very much. Now, the first question I would like to know is what exactly is biochemistry? Because a lot of people wonder if it's just a mixture of both biology and chemistry, and to what extent.
Yes, so we mix both biochem and chem. So what happens is that we look at biological molecules and look at the chemistry behind it. So if you think about enzymes, they go through a series of reactions almost like chemistry, but we look to see how they do it.
Okay, so let's look at the mechanisms.
Yeah, so a lot of mechanisms and how they work together for the whole, let's say, any animal or any organism. So down to a bacteria, up to as big as an elephant, we can look at.
As I know, biochemistry is not taught at school, and what age did you decide, yes, it's biochemistry?
I've always been interested in science. I think that started in elementary school. The drive to do biochemistry led for my interest in biomedical research. So that kind of came along, I would say, in my early 20s. I would have gotten a lot of research projects in that kind of area, and then I kind of veered into it after that.
So speaking of research, because you say you've done a lot of research, what research are you currently working on?
So my area of research is biomedical, but my specialty is diabetes and obesity. So we look at how diabetes and obesity relate to changes in your vascular system. So all the vessels and arteries, why in particular they're clogging, why in particular they're thinning, why in particular we have an issue. So we look at biomarkers to stage a disease. So if you think of diabetes, we think about the amputation, the foot. And then we have to understand, well, what's going on that drives you from having a healthy foot to an amputated foot. So we look to see all the stages that happen in between there.
Which markers are generally present? Which markers do you actually look for? The DNA markers, or is there a special type of marker in the body that will tell you?
Okay, so when we say markers, we're normally talking about, it could be a range of, it could be DNA, RNA, and it could be just protein. So we have recognized that when you are in a particular state of a disease, different proteins get increased and different proteins get decreased. So like if you know you're having an allergic reaction, you know all your histamines are going to come about and give you all the symptoms. Same thing. So for diabetes, we know that insulin's an issue. So we're going to look to see the changes of insulin, and then we're going to look for any other proteins, let's say, that are going to fluctuate. So that's what we do. We look for changes in proteins, and then we know that specific proteins are associated with that disease state.
Okay, so it's just like you do not just look at the insulin levels.
No.
You look at the factors that
everything. So we can look at fat, we can look at sugars, we can look at inflammatory responses. We look at a whole slew of markers. I think when we first started, we started out with 300 markers. We've actually narrowed it down to about 50, which gives you a full picture, because we can actually pinpoint if you're early in the disease and what you look like in the later stage of the disease.
Saying that you've done all this research into diabetes, you have definitely met some interesting scientists along the way. Which one is the most standout scientist to you?
Oh, there's several. As a female scientist, I think I would have admired Professor Catherine Cienfone. She actually started out in cardiovascular research, and then she became fairly large in North America. And then Dr. Alan Seidelman, he would have been a cardiologist who has bridged the gap between cardiology and obesity. And then last but not least, I think he's the last one that was fairly prominent, is Carlos Ferrero. He's at the Wake Forest Medical Center, and he was looking particularly at how we manipulate salt, and he was able to show there's a difference in let's say, people of African descent versus people of non-African descent. And that became very important to my work because, in the Caribbean, being of African descent, we're seeing various differences in how we metabolize even glucose. So it was nice to work with him because we were actually able to kind of bridge two areas that were never bridged before.
Those are international scientists. Yes. So now, are there any local scientists or upcoming scientists that you really have like your hopes on? Like you could say, oh, that person is going to be well known in their field of study?
I think because the Caribbean, we're so junior in a lot of our work. And then we do have some big players. Like we do have some big players coming out of Jamaica. We do have some big players coming out of St. Augustine. So that would have been Trinidad and Tobago. However, their area of research, it's not really my focus. So I'd have to say I haven't really met some big players in my area. But it doesn't mean that they're not junior persons that I haven't met as yet that are now making great strides.
Speaking of that fact, where do you see Caribbean scientists really excelling in the future?
One, we have to be able to use our resources more effectively because we have very little. So we really do have to get together a lot more. But I do see us pushing toward the biotechnology. We have the capacity to do biotechnology very well. And I think if we can keep up with that, then I think we would be well known in the international forum. But it really comes with the biotechnology.
Biotechnology, can you go into a bit of detail about what exactly biotechnology is?
So it's the application of almost like biochemistry. So we can manipulate a microorganism that is going to, clean the water without harming the person who may drink it. So we can manipulate things. We can go in, affect genes. We can ask, for example, we can produce insulin from a bacteria. Yes. Before we couldn't do that. So we had to engineer that bacteria to say, I need to produce insulin. I need to produce it when you tell me to produce it. So it's things like that. Producing things that can help our environment, producing things that can help in our processes. So whether our processes are in manufacturing, whether our processes are cleaning up the environment, or whether they're in producing a product, that's what we need to do.
Yeah. Oh, back to the bacteria to produce insulin. A few years ago, a child was doing some research on diabetes, and they brought up a fact that there's wheat that there's some other organism, I can't remember what it was like wheat, I think, that they're now using to produce insulin from. Is there any truth to that? Because when she had said it, I was just like, this is new science to me. Is that
so what they would have done is they would have done a kind of a fusion with a plant. Now the problem that you're going to face, it will produce a form of insulin, but will it be the form that our bodies can actually use well? And that's always the issue. So we always have to recognize that anybody can put anything in anything, and you can get any organisms produce that protein. But if it's not an animal-based structure, then how do you make sure that the animal who wants to take it, like us, will benefit from it? And that's one of the problems we face with insulin. Before we could do it in a bacteria, we had to do it in yeast, because yeast is closer to a human cell than it is to bacteria. And if we don't fold a protein properly, it's no good to us. So it comes about with all the fine details of getting it to the point where it becomes beneficial.
Okay. Could you explain to them what exactly you mean by folding protein?
Okay, so let's say we have a word, and you spelt my name. Okay, so MANNING. But the body doesn't see it that way. The body sees it all crumpled up in a certain conformation. And if it's not in this conformation, then it's not recognized. It can't fit. It can't fit the signal. So it has to meet something, and it has to fit and send the signal. Now, if it was in this structure and it went to do like that, it doesn't fit. So that's what we talk about folding. It has to go from a linear or a straight line to a very complex folding, and that allows us to then send all the little signals we need to get your body to work properly.
And it's because of this folding that enzymes are
the major players, yeah. For example, your immune system.
mm-hmm.
Your immune system works through a series of enzymes that activate another series of enzymes,
mm-hmm.
which at the end turns on your DNA, which at the end tells the cell to do two things. Either you need to go and die, or you need to live.
Let's not let that not.
Well, that's a good thing. Imagine if it's a cancer cell.
Oh,
we want the cancer cell to die.
Oh, that's what you mean by saying let's go die.
Yeah, let's go die, or we say let's go live. So the cell has to make a decision based on those proteins that came and told it what to do.
Yeah. Okay. Yeah. So that's individual cells in the body and not the whole body saying
no. But you do know that our body, the cells in our body die, and then we regenerate every couple of days, right?
Yes, I do know that.
Okay.
What improvements would you make to our educational system so that science will be more appreciated? Like, yeah.
Can I be honest?
Yeah, sure. Because honestly, I personally think like you don't meet the cool science until you come to university or.
Well, besides the money, I think we need to have people teaching kids that enjoy the science themselves. I think when you have teachers that don't enjoy the science, it gets a little boring because they're not putting their all into it. And it's not their fault, it's just that they're not interested.
Yeah.
And I honestly think that we need to come away from the books. I think we have to do a lot more hands-on. I think that's how we grow. That's how we come up with these grandiose ideas, and we have to come outside the box. I think we really have to think outside the box to really push science forward. So I really think for schools, you really do need a teachers who are excited about science. And so then that requires proper training and the love of science. And then besides the money, you really do need to do more hands-on. And hands-on can be simple things, but give a student something to do and tell them to solve it. And don't Google it. Solve it. Come up with something. And praise them for the fact that they thought outside the box.
Yes.
And we don't do a lot of praising in the Caribbean. I think that's one of the problems I'm seeing, even with, because I have a child in secondary school, and I see it when he talks to his friends. It's so different when they feel that they're going to be judged negatively when they should just be told, hey, the fact that you thought something is a beginning.
Yeah.
Yeah.
Because that's all science starts with. It's just like, I wonder if this can work.
Yes. Yes.
Sometimes it may work and sometimes
it
it doesn't.
And that's the worst thing. If it doesn't work, it's the worst thing, but no big deal. You know what? You move on. Because if you didn't have that thought, then you didn't have the other thought, and the other thought can bring you something that is amazing that works.
Yeah. And the last question now for you is, what would you say to our young viewers who are interested in science but feels a bit
overwhelmed?
I don't really want to say overwhelmed. I mean, like as we pointed out, that science in school is not as fun until you get to the later stages. So what would you tell them to keep them motivated to do science?
Oh gosh, don't give up. If you're interested in something, follow it. Talk to people about it and keep your spark, because it's only Barbados. And I say that in a way that the world is huge, and no longer are we functioning in our own little world, we're functioning outside our world. So don't think because you don't see it here that it can't happen. They can go anywhere. The world is their open oyster. And I think that if they keep positive, they can become anything and do anything and solve even the world's biggest problems. As I tell all my students, you are the next Nobel Prize winner, so hurry up and make me proud.
Yeah, don't you want to be a Nobel Prize winner?
I would love to, but I also know that the amount of money that I have brought in, I cannot get there that far. But I still have that dream. So hopefully in the next 10 years, I may actually obtain a Nobel Prize.
Yeah, so we should all be paving the way for the next scientists to come along.
We should be.
Don't worry that we can't do it ourselves in our time, but maybe the next generation or the generation afterwards.
Most definitely.
Yeah. So there you have it. This has been Dr. Thea Scantlebury-Manning, who is a biochemistry lecturer at the University of West Indies, Cave Hill. And again, I thank her for being on this show and hope to hear great things in the future.